Hello and good evening, everyone. So, how are you guys doing today? Yes. Okay. So, welcome to another live session on Eden 2. Now, today's live session is exclusively for our NEET MDS aspirants, especially for the 2027 batch. Okay. Uh, According to our schedule, after completing biochemistry, now we are moving on to microbiology. Okay. So, as microbiology is it's your second year subject, and yeah, so today is the first live session of microbiology. So in today's session we will discuss in detail about some of the important concepts from general microbiology and immunology. Okay, so in the initial part of the presentation, I'll be discussing in detail some of the important concepts from general microbiology and immunology. And then towards the end, I will discuss some of the important MCQs. Okay, some of the MCQs from the previous year question papers also. Okay, so that you will get an idea about the type of questions that you can expect for your NEET MDS examination. Okay, so yes, let's get started. So it's a second year subject. It's mostly a laboratory based subjects. Okay, so there are like lots of factual data and names of the medium like that and all to remember. Uh, if you look at the initial few NEET MDS examinations, you can see that like from microbiology, more and more questions like direct, straightforward, or factual based questions were frequently asked. Okay, in the initial few years of the NEET MDS examination. But if you look at the recent years, you can see that application based or clinical based questions were asked even from such lab based subjects like general microbiology. Okay. Not much, but yes, you can expect application based or clinical based questions from microbiology also. Okay. And especially after the COVID period, there were more questions from sterilization and disinfection and related to COVID virus also. Okay. So have a look at those topics also. Okay. So before going into a session, let me introduce myself. I am Dr. Nivedita Tampan. I am the co-academic director of Eden 2 and your mentor for microbiology. Okay. Yes. So first, let's have a look at the weightage of microbiology subject. Okay. So according to the official website of MBMS, you can expect a total of 14 questions from both general pathology and microbiology combined. Okay. So from microbiology alone, you can expect somewhere around like 6 to 7 questions. Okay. If more questions were asked from general path, then there will be less questions from microbiology and vice versa. Okay. That can happen. Okay. But usually you can expect somewhere around like 6 to 7 questions from microbiology alone. Okay. And let's see which are the overlapping subjects with microbiology. So it's very much like combined with general pathology. All the initial chapters like the immunity, hypersensitivity reaction, the immunodeficiency diseases, and the infections, all those topics are there in general path also. Okay, so while studying, try to study the club these topics together and study. Okay, so that you will be able to cover a lot of topics at one go. Okay. And other than that, the infections topics. Okay, the infections topic is coming under oral medicine, under oral path, under general medicine also. Okay, so all these infections can be the bacterial, viral, and fungal infections. They are like a little bit similar. to what we study in the general microbiology. Okay, we are more focused on the clinical features in medicine and pathology and all. But yes, the mm, type of organism and how it can be cultured, that and all we are studying under this microbiology. Okay, so there is like relation or overlapping topics with these subjects also. And the periodontal microbiology, yes, somewhat overlapping topics with that also. Okay, so these are all some of the overlapping subjects with microbiology. Yeah, I will suggest you to club these subjects together and study so that it will be much more easier for you, and you will be able to cover like a bulk amount of topics at one go itself. Okay. Yes. Now, which all books you can follow? I will suggest you to follow the book that you had learned in your UG days. Okay, in your second year BDS, you must have studied or like revised or learned from a microbiology textbook, right? Follow that itself. Now, other than that, you can also follow CP Baweja and another Narayan and Panikar's textbook of microbiology. Okay, NEET MDS exam is a MCQ based exam, so MCQs must be given more importance. But if you want to understand some concepts in depth, or you want to understand some concepts thoroughly, then only refer to the textbooks also. Okay, so Narayan and Panikar is like very elaborate compared to Baveja. Baveja is somewhat concise and to the point. Okay, so whichever textbook you have, you can refer that. All right. Now, next, let's have a look at the sure short chapters and the cold spot chapters. Okay, so what do you mean by sure short chapters? So after analyzing like for the past like 10 to 15 years of the NEET MDS question papers, there are some topics from where questions are like frequently repeated every year. Okay, the questions are not repeated as such. The questions will be definitely different, but these topics, okay, the topics are important. Okay, because from these topics the questions were very frequently asked. Okay, so that is the sure short chapters. Then we have the cold spot chapters also. Cold spot chapters means they are important, but questions may or may not be asked from these chapters. Okay, so they are called as the cold spot chapters. Now, which are the sure short chapters? Sterilization and disinfection. Very important. Culture, media and methods, antibodies and immunoglobulins, hypersensitivity reaction, and under bacteria, staphylococcus and streptococcus, extremely important. And other than that, you should also have a look at mycobacterium and actinomycetes. Okay, Clostridia, Conibacterium can be asked. Okay, virology, HIV and herpes, extremely important. Hepatitis, MMR. Okay, the mumps, measles, and rubella can also be are also important. Coming to mycology, candida and mucormycosis are extremely important. And you can also expect questions from immunodeficiency diseases and sometimes from Vibrio cholerae and the spirochetes, Treponema pallidum. Okay. Virology, yes, COVID virus is important. Mycology, again, Aspergillus and Histoplasma can be asked. Okay. Parasitology, malaria, and entamoeba histolytica can also be asked. Vaccines. Okay, vaccines are another important topic, but not repeated every year, but yes, it is important. Okay, so these are some of the sure short chapters and cold spot chapters. Before going into the exam, do not miss these sure short chapters. Okay, those are like extremely important. Do not miss those sure short chapters at all right. Yes. Now, what type of questions can you expect from NEET MDS examination for microbiology? You can expect three types of questions. Okay, there are like direct, straightforward questions, just like this. Okay, McConkey's medium is what? Okay, this McConkey medium question is the one that was asked in NEET MDS 2024 examination. Okay, so they can ask such direct, straightforward questions, or you can expect image based questions also. Okay, they can give an image like this and ask you to identify what is given in this image. Is it a complement fixation or IgM, or is it like antibodies like immunoglobulins like M or A, or antigen antibody complex? Yes, such questions can also be asked. Then clinical based questions. Okay, the clinical based questions are not as frequently asked as like from other clinical subjects, but yes, you can expect clinical based questions also. Okay, like a patient with history of COVID-19 reported to the OPD with maxillary necrosis. Okay, on histological examination, both septate and aseptate hyphae were seen. Which microorganism can it be? Can be aspergillus or mucor. Like that, questions can be asked. Okay, so nearly like 50 to 65 percentage of the questions will be like direct straightforward questions only, but the remaining 20 percentage they can ask image based and clinical based questions. Okay, so whichever way they ask the question, you must be thorough regarding your basic concepts. Okay, basic concepts. And if you are like thorough regarding your basics, then it doesn't matter how they ask the questions. Okay, you will be definitely able to answer it. Okay, so strengthen your basics first. Okay, so let's first discuss some of the important basics from general microbiology. Okay, so starting with sterilization. What is sterilization? It is the process by which all the articles or surface or medium is freed of all microorganisms in both vegetative and in the spore state. Okay. Spores are difficult to eliminate. Okay. The bacterial spores are like really difficult to eliminate because they are resistant. Okay. They are really resistant in nature. Okay. So the sterilization is the process in which they are able to eliminate even these spores. Okay. So the sterilization can be done with the help of two types of agents. Okay, there are like physical methods and chemical methods are there. Okay, we'll be discussing only the important ones, not everything in detail, the important ones that are needed for NEET MDS examination. Okay, so the physical methods can be with the help of heat or filtration or radiation. Okay, so this heat can be of two types. There is dry heat and moist heat. Dry heat includes like flaming incineration and hot air oven. And moist heat, the most important method is autoclave. Okay, so in this heating method, mainly what is happening is that the protein of the cell, okay, of the bacterial cell is getting denatured. Okay, so mainly protein denaturation and coagulation is happening. Okay, so that's how the cells are getting destroyed. The protein is getting denatured and the protein is getting coagulated. Okay, so that's how the bacteria is getting destroyed. Okay, then there is something called as radiation. Okay, we can also use radiation for destroying the bacteria and its spores. Okay, so there are two types of radiation again. There is non-ionizing radiation and ionizing radiation. Non-ionizing radiation include infrared and UV rays, whereas ionizing radiation includes X-rays and gamma rays. Okay, among this gamma radiation is gaining popularity nowadays. Okay, like lots of the surgical instruments and the surgical gloves and all those things are like mainly done by the sterilization is mainly done by gamma rays only. Okay, then there is something called as cold sterilization. Cold sterilization means here the temperature is not at all increased. Okay, if you look at the heating method of sterilization, the temperature of that particular medium is getting increased. Okay, but there are some substances that are like sensitive to heat. Okay, so we cannot like heat such materials. Okay, the materials that are made with plastic. Okay, or the materials that are not heat resistant. Okay, we cannot just heat them. Okay, so what we can do is other methods of sterilization, which includes the cold sterilization. In cold sterilization, what they are mainly doing is that the temperature is not at all increased. So all these methods like the filtration and the radiation methods of sterilization, they all come under this cold sterilization method. Okay, because the temperature is not at all getting increased. Okay, so that is called as the cold sterilization. Now, let's have a look at the most commonly used moist heat method of sterilization. That is the autoclave. Okay, autoclave is it is also called a steam sterilizer, and it is the one that is most commonly used in almost all the dental clinics and all. Okay, so let's have a look at the structure of the autoclave. There will be uh, it is just like a modified pressure cooker. Okay, there will be a stainless steel container, and it will be supported by an iron case, and with the help of electricity, the water will be heated, and the steam will circulate throughout, and it will supply under high pressure to the inner chamber also. Okay, where the instruments are kept. Okay, so this will result in sterilization of the instruments. Okay, and in the upper part there are like release valve and safety valve. All those structures will be there, and the pressure gauge will be there to help us see how much the pressure is getting increased. Okay, and all those instruments will be there. Okay, this temperature and the holding time for autoclave is important. Okay, now in some of the previous examinations, they asked about this, the temperature and the holding time. Okay, so the normal temperature that we set for autoclave is 121 degrees Celsius, and the holding time is 15 minutes. Okay, so that is the normally. If we are increasing the temperature, then the holding time can be decreased. Okay, so if we are increasing the temperature to 126 degrees Celsius, then the holding time can be decreased to 10 minutes. If we are again increasing the temperature to 134 degrees Celsius, then again the holding time can be decreased to just 3 minutes. Okay, so the temperature and the holding time is important. Please go through it. The sterilization conditions, okay, for the autoclave is important. Now, after the physical agents, let's have a look at the chemical agents that we use for sterilization. Okay, so chemical agents, they are like mainly employed in antisepsis and in disinfection only. Okay, they do not mainly affect the spores and all. Okay, they are mainly used as antiseptics and disinfectants only. There are alcohols, aldehydes, phenols, and halogens, oxidizing agents, metallic salt, and other vapor phase disinfectants. Okay, so many chemicals can be used as disinfectants. Among these, the alcohols and aldehydes are really important. Okay, so the alcohols, which all alcohols can be used? We can use the ethyl alcohol or isopropyl alcohol or the methyl alcohol. Okay, all these alcohols are mainly used as skin antiseptics only. Their main function is to denature the bacterial proteins. Okay, again, here also they are denaturing the bacterial protein. Since I had already mentioned, they do not have any action on spores. Okay, they are not that strong enough to destroy the spores. Okay, sometimes this methyl alcohol can alone like attack the fungal spores. Okay, but otherwise ethyl alcohol, which is the most commonly used one, it is mainly used as an skin antiseptic only. Okay, it cannot destroy any spores. Okay, then we have the aldehydes. The common ones are like formaldehyde and glutaraldehyde, which has like bactericidal, virucidal, and sporicidal activity. Okay, it can kill the bacteria, it can kill the virus, and it can even kill the spores. Okay, even like formaldehyde can kill the anthrax spores, and glutaraldehyde also. Okay, against the tubercle bacilli, fungi, and viruses and all, it can be used. Okay, then comes this oxidizing agents. Okay, so these oxidizing agents are also important because here comes the hydrogen peroxide and peracetic acid and plasma sterilization. Okay, so what do you mean by plasma sterilization is that mainly an electromagnetic field is created. Okay, so into this electromagnetic field, we are adding the hydrogen peroxide vapors. Sometimes the hydrogen peroxide vapors alone, or sometimes along with this hydrogen peroxide vapors, we are adding the peracetic acid also. Okay. And these vapors, they create a state of plasma. Okay. Once they create this state of plasma, what happens is that free radicals, like the uh, hydrogen free radicals and the oxygen free radicals, they are released. Okay, so these free radicals has the ability to go and attack the cells. Okay, so they are highly reactive in nature. So what they does is that they will just readily go and attack these bacterial cells, and they will denature all the microbes. Okay, so that is called as plasma sterilization. So in this plasma sterilization, we are mainly used either hydrogen peroxide or peracetic acid vapors. Okay, so the plasma sterilization is also gaining more importance nowadays. All right. Yes, so that's about the chemical agents. Now, this is like a summary of all the methods, and which all materials can be sterilized by which methods. Okay, so uh, other than what is given here, okay, this is like a general table. Okay, they had included like culture media, cytoscope and endoscope and all. In addition to that, you should also know how the dental materials like the aerotos or dental burrs or the other materials are getting sterilized also. Okay, some of the important things like the gloves, the surgical gloves, the glasswares, and the disposable syringes, how they are sterilized is like very frequently asked questions. So please go through that once, go through this table also. All right. Now there is something called as sterilization control. Okay. Sterilization control means we are checking and confirming whether the sterilizer is actually working and it is like killing all the microorganisms. Okay, so that is what is called as sterilization control. So for checking for the sterilization control, or for checking the effectiveness of this sterilization, we are using three methods. Okay, there is like physical method, chemical method, and biological method. In physical method, we are using like temperature gauge or pressure gauge. Okay. When we look at the temperature and we are analyzing temperature and we are analyzing the pressure and predicting that, okay, the sterilization has been been completed. Okay. Then there is some method called as chemical methods. Okay, where we are using indicator strips. Okay, so these indicator strips will change color, and we will come to know that, okay, the sterilization is complete. Okay, and the most reliable one is this biological method. Okay, so in this biological method, what we are doing is that we are placing some highly resistant bacterial spores inside the sterilizer. Okay, near to the instruments. Okay, we are placing some highly resistant bacterial spores inside the sterilizer, and at the end of the sterilization, we are checking whether these spores are present or not. Okay, so if these spores are killed, then that means that the sterilization is successful. Okay, but if the spores survive, then that means that the sterilization has failed. Okay, so for different methods of sterilization, we use different types of spores. Okay, so the one that is most commonly used is Bacillus stearothermophilus. Okay, for the plasma sterilization and for autoclave method, we mainly use this Bacillus stearothermophilus only. Okay, but if it's an ionizing radiation, then we are mainly using like Bacillus pumilus. Okay, if it is ethylene oxide sterilization, then we'll be mainly using Bacillus atrophius. Okay, so this is a summary of the sterilization method and which biological indicator organism can be used. Okay, so the commonly used one is Bacillus stearothermophilus for autoclave, and for the plasma sterilization, we use the Bacillus stearothermophilus only. But for dry heat and ethylene oxide, we use Bacillus atrophius. Okay. And for the gamma radiation, we use mainly Bacillus pumilus. And for chemical sterilants also, we mainly use Bacillus stearothermophilus. Okay, so like formaldehyde and all, glutaraldehyde and all also, we use Bacillus stearothermophilus. Okay, for the dry heat and ethylene oxide, and for the gamma radiation, it varies. Okay, for all the other methods, it is Bacillus stearothermophilus only. Okay. So just like we had like sterilization control, there is like testing the efficiency of disinfectants also. Okay. So disinfectants can be tested with the help of like four tests are there. One is radial Walker test. Then you have the Chick-Martin test. Then the Kelsey-Sykes test is also there, and in-use test is also there. Okay. So among these four tests, this radial Walker test is more important. Okay. So what they are testing in this radial Walker test is that they are checking the ratio of dilution of any test disinfectant. Okay, whichever disinfectant we want to test, we are checking the ratio of that particular disinfectant with that of the dilution of phenol. Okay, at the same time and under identical conditions only. Okay, so we are checking the ratio. We are checking the ratio of this test disinfectant with that of the phenol. Okay, so we'll get a number that is called as the phenol coefficient. Okay, so if this phenol coefficient is greater than 1, then that means that this particular disinfectant that we had tested is effective. Okay, if it is less than 1, then that means that the phenol coefficient is not effective. Okay, so this is how we test for disinfectants. Okay, so among this, the radial Walker test is extremely important. Okay, so that's all about the sterilization and disinfection. Now we are moving on to culture media. Okay, so culture media means bacteria can be grown in laboratories. Okay, we can study bacteria, we can grow bacteria inside the labs, and we can study them. Okay, so these bacteria need like some special environment for it to grow. Okay, so such kind of special environment that is required for the bacteria, or special food that is required for the bacteria, that is called as the culture media. Okay, so if you want to grow a plant, you need a fertile soil for the plant to grow, right? So just like that, this if you need the bacteria to grow, then you need a like a particular media. Okay, that media is called as the culture media for the bacteria to grow. Okay, so just like how the soil helps in the growth of the plant, just like that only how the culture media acts for the bacteria also. Okay, so it's like a fertile soil for the bacteria, this culture media. Okay, so this culture media can be of many types. There is like simple, complex, there is like liquid, solid, and like synthetic media, and like so many definitions of culture media are there, but based upon the functional requirements. Okay, so based upon the functional requirements, this culture media can be of 6 types. Okay, there is enriched media, there is enrichment media, selective media, indicator media, differential media, and transport media. Okay, so these are the 6 types of culture media. Okay, so while making a culture media, the base will be usually same for almost all the culture media. Okay, so the base will be usually made with agar. Okay, then to this agar, you will be adding like blood or egg or serum for the growth of the bacteria. Okay, so if you are adding such constituents like blood or egg or serum to the basal media, then that becomes enriched media. Okay, so these examples of enriched media is blood agar or chocolate agar, like that. Okay. Then there is something called as enrichment media. Okay. So this enrichment media means, suppose we are taking a swab from our oral cavity. Okay. And we only want to grow the Streptococcus mutans. Okay. But in the swab that we had taken, since oral cavity has all types of bacteria, there is like Streptococcus salivarius, there is like other types of all the microorganisms will be there, like lactobacillus will be there, all the organisms will be there. But we only need Streptococcus mutans. Okay. So if you are using an enrichment media, then what it will do is that it will suppress all the commensal bacteria. Okay. So it will suppress all the commensal bacteria. Okay. So commensal bacteria like maybe lactobacillus or salivarius is suppressed. Okay. So if their growth is suppressed, then which bacteria is growing? Only Streptococcus mutans. Okay. That is the bacteria that we need. Okay. That is the bacteria of interest. Okay. So such media is called as the enrichment media. And the examples are like tetrathionate broth and the selenite F broth. Okay. It will, this tetrathionate broth and all, it will allow only the typhoid bacteria to grow. Okay. And suppresses all the other types of bacteria. Okay. Then there is something called as selective media. Okay. Selective media means the greater number of required bacteria, it forms colonies. Okay, so in the enrichment media, what it does is that it is suppressing all the commensal bacteria. But in selective media, what it does is that it is only allowing the growth of the bacteria of interest. Okay, again, we are taking a swab from our oral cavity. It has all types of bacteria, mutans, salivarius, lactobacillus, all types of bacteria will be there. Okay, our bacteria of interest is only mutants. Okay, so if we are using a selective media for mutants, then only the Streptococcus mutants will grow. Okay, others won't grow. Okay, so that is called as the selective media. Okay, so there are like many types of selective media like disoxycolate citrate agar, thiosulfate citrate, bile sucrose agar, and so many selective medias are there. Okay, then there is something called as indicator media. Indicator media means when a particular bacterial colonies are growing, then that particular culture media will change its color. Okay, the if the mutants is growing in that particular petri dish, then it will start changing the color. Okay, so when it changes the color itself, without looking at it under the microscope itself, we can predict that, okay, so this bacteria is growing in this petri dish. Okay, so that is called as the indicator media. Okay, there are so many types of indicator media like the MacLeod's medium or the Wilson-Blair medium, like that. So many types are there. Then we have the differential media. Differential media is used to differentiate one group of bacteria from the other group. Okay, so differentiate media mainly like, for example, McConkey medium. Okay, if you look at the McConkey medium, it is used to differentiate between lactose fermenters and non-lactose fermenters. Okay, lactose fermenters will produce pink color colonies in this McConkey medium, and non-lactose fermenters, they will be like pale colored or like light yellowish colored in McConkey medium. Okay, so this is a medium that is used to differentiate between two types of organisms that are growing. Okay, so that is called as the differential medium. And this McConkey medium is also important because it had been previously asked. Then the transport medium. Okay, from the surgical department to the lab, we need to carry these specimens in a particular transport medium. Okay, so the commonly used transport medium is Stewart's medium. Okay, this is called as Stewart's medium. So let's have a look at this McConkey medium because it had been previously asked. So this McConkey medium, it is made with peptone, lactose, agar, and neutral red and chloroform. Okay, so like I had already mentioned, it is used to differentiate between lactose fermenters and non-lactose fermenters. Okay, lactose fermenters, the example is like E. coli. E. coli will produce pink colonies like this. in the McConkey medium. Whereas non-lactose fermenters, it will produce like colorless or pale colonies like this in the McConkey medium. Okay. Examples are like Shigella and Salmonella species. They are like non-lactose fermenters. They will produce such colorless or like pale yellow colored colonies like this. Okay. So it can be considered as an indicator medium also, because based on the color, we can predict where the which organism is growing here. Okay. So yes, it is a differential medium and can be considered as an indicator medium also. Okay, because it is changing color when the organisms are growing. Okay. Now, like I had already mentioned, the commonly used transport medium for most of the specimens is Stewart's medium. Okay. Now, there is like a special medium, transport medium that we use for IHC, okay, immunohistochemistry and immunofluorescence studies. Okay, where you does your immunohistochemistry and immunofluorescence studies like that, mainly for vesiculobullous lesions. Okay, like pemphigus vulgaris, lichen planus, or like SLE and all, we do immunohistochemical studies. Okay, so these specimens cannot be transported in a normal medium or in a Stewart's medium. Okay, because all the enzymes can destroy its immunoantigenicity. Okay, so you need a special medium for transporting this uh, tissues that are indicated for immunohistochemistry. Okay, so which medium is used for transporting such specimens? Mainly Michel's solution. Okay, it is the Michel solution. Okay, so this Michel solution contains the magnesium sulfate distilled water and ammonium sulfate and the KOH also. It is the transport medium that you use for transporting immunohistochemistry and immunofluorescence studies. Okay, the tissues that are indicated for immunohistochemistry and immunofluorescence studies, yes, mainly you use Michel's solution. Okay, why Michel solution? Because it preserves that immunoantigenicity and it also inhibits the proteolytic enzymes. Okay, so that's why you are using this Michel's solution. Okay, so this is regarding the two types of transport media that we can use. Normally, Stewart's medium, and for IHC studies, we can use Michel's solution. Now, next, so far we had looked at the aerobic culture methods. Okay, so bacteria can be of two types. There is like aerobic bacteria and anaerobic bacteria. Okay, aerobic bacteria means the ones which can survive in oxygen. Okay, the ones that need oxygen to survive. Okay, so such bacteria are called as the aerobic bacteria. Whereas anaerobic bacteria means it oxygen is like toxic to them. Okay, it does not need oxygen. Okay, it like survives better without the oxygen. Okay, so such bacteria are called as this anaerobic bacteria. Now, this anaerobic bacteria cannot be like cultured just like we culture the aerobic bacteria. Okay, it's much more difficult and like advanced methods must be used to culture these anaerobic bacteria. Okay, so for anaerobic culturing, we use special methods like this Macintosh and Filldays anaerobic jar, gas pack system, re-reduced anaerobic system, anaerobic chamber or glove box, and anaerobic culture medium. Okay, so these are some of the methods by which we can like culture anaerobic bacteria. Okay, so this Macintosh and Filldays anaerobic jar is the most frequently used one. There is a vacuum pump that is connected at the top of this jar, and what it does is that it will evacuate all the air inside and supply it with hydrogen gas. Okay, that will result in the growth of this anaerobic bacteria. Okay. Then there is like gas pack system. In the gas pack system, there are some disposable envelopes that are present like this. Okay, so these disposable envelopes, it will generate hydrogen and carbon dioxide. When you add water to it, it will help in the generation of hydrogen and carbon dioxide. Okay, and it will create that anaerobic environment. Okay, so that is called as gas bath system. Then in pre-reduced anaerobic system, again, already prepared media is used. Then in anaerobic chamber or glove box, it is like a airtight chamber. Okay, it is like an airtight chamber and it is filled with inert gas. Okay, inert gas means the gases which are like chemically not reactive. Okay, they are stable. Okay, and they do not readily undergo any chemical reaction. Okay, so such gases like helium, neon, all those gases are added to this anaerobic chamber. Okay, so that is about the anaerobic chamber. And anaerobic culture media is also there. Okay, so this anaerobic culture media, it includes three main media. One is Robertson's cooked meat medium, thioglycolate medium, and Smith-Noguchi medium. Okay, so these are the three anaerobic culture media that is present. Okay, so this in this Robertson's cooked medium, which is the most commonly used one, it is like a transport media also. It has like fat-free minced cooked meat. Okay, like cooked meat like this will be present, and the meat will change color when the organisms grow. Okay, the meat will change color to either pink or red or blackish in color. Okay, so that's about the Robertson's cooked meat medium. Then we have thioglycolate medium. Thioglycolate medium, it contains like extracts of yeast and acetone, sodium chloride, cysteine, thioglycolic acid, and methylene blue, and all these are mixed together. Okay, so since so many ingredients are there, the oxygen at this thioglycolate media will be highest at the top. Okay, so if you look at here, the oxygen concentration will be high here and it will be low here. Okay, so since the oxygen concentration is high at the top, the aerobes will grow at the top. Okay, and the anaerobes, they will grow only at the bottom like this. Okay, why bottom? Because the oxygen concentration is less at the bottom, so they will grow only at the bottom. Okay, whereas the oxygen concentration is high at the top, so only the aerobes will grow at the top. Okay, these anaerobes, they will grow at the bottom only. Now there is like a third category that is called as facultative anaerobes. Facultative anaerobes means they can survive in oxygen and they can survive without oxygen also. Okay, so such facultative anaerobes, they will be spread throughout the medium like this. Okay, you will be able to see such organisms throughout the medium. Okay, so based upon at which position the organism is growing, we can predict whether they are aerobes or anaerobes in thioglycolate medium. Okay. Then there is something called a Smith-Noguchi medium. The Smith-Noguchi medium is mainly used for cultivation of spirochetes, like the Treponema pallidum. Okay. Mainly indicated for Treponema pallidum. Okay. So that's about the culture media and the anaerobic culture media and the anaerobic culture methods. Now we are moving on to immunity. Okay, so what do you mean by immunity? Immunity means it is the resistance by the host towards any outside microbes. Okay, so the immunity is of two types. We have innate or native immunity and acquired or adaptive immunity. Okay, so this innate immunity, it will be usually present at birth and it is non-specific. Okay, it is not specific against any bacteria or any virus like that. Okay, it is like non-specific. It is like active against all the bacteria and all the virus like that. Okay, and it is immediate in nature. Okay, when they attack, this immunity reacts. Okay, it is like non-specific but immediate response will be given. Okay, so that is called as innate or native immunity. It can again be classified into two types, non-specific and specific, and it again includes a subcategory of species immunity, racial immunity, and individual immunity. Okay, so we as a human species, is not susceptible to any plant pathogens. Okay, the ones that affect the plants, they cannot affect us. Okay, so we are like innately immune against such pathogens. Okay, then there is like racial immunity. Okay, um, some race are like less prone for tuberculosis, whereas others are more prone for tuberculosis. Like that, there is like racial differences between the in among the human race itself. Okay, then there is individual immunity also. Like every person's immunity is different. Okay, some persons have like high immunity, some persons have like less immunity. It's based upon a lot of factors like age, gender, or like nutrition. It's based upon lots of factors. Okay, then we have the acquired or adaptive immunity, which usually develops after exposure. Okay, so after exposure, what happens is that the body will uh, keep a memory of the antigen that has attacked it. Okay. Now, when the same antigen attacks us, a secondary response is given by the body, and the secondary response will be more strong than the primary response. Okay. So it will be more specific and it will be more strong, okay, than the primary response. Okay. So that the second time when the same antigen comes and attacks us, the antigen will be like completely eliminated from the body. Okay, so that's called as acquired or adaptive immunity. Okay, this acquired or adaptive immunity is again divided into two types. We have the active acquired immunity and the passive acquired immunity. Again, this active and passive is again divided into natural and artificial. So natural active immunity is post-infection. Okay, uh, like chicken pox. Okay, chicken pox virus, once it uh, attacks a patient, usually the second time attack is not usually seen. Okay, so because they develop these antibodies against it. Okay, then we have the artificial response. Okay, so this artificial active immunity, the vaccines. Okay, the vaccines are the example of artificial active immunity. Then the natural passive immunity is the immunity that is given from mother to child, okay, through the colostrum and through other means also. Okay, so that is called as the natural passive immunity, and natural artificial passive immunity will be there in the form of antibodies. Okay, so this passive immunity is mainly the antibodies which comes from an external source, and the active immunity, the body produces its own antibodies. Okay, so active immunity is much more powerful than that of the passive immunity. Okay, so this artificial active immunity is vaccines, and it is the immunity that is induced by vaccines. Okay, so what are vaccines? Vaccines are like any preparation of like live or killed microorganisms, or it can be the organism as a whole, or it can be any product of the organism also. Okay, it is used for immunization. Okay, so based on that, there are like five types of vaccine. We have the live attenuated vaccine, inactivated vaccine, subunit vaccine, toxoid, and the latest mRNA based vaccines. Okay, so this live attenuated vaccine means we are taking the microbe and we are weakening it. Okay, not killing it. We are just weakening it. Okay, not killing it completely. Okay, we are just weakening it and making it as a vaccine and injecting it into the humans. Okay, so what happens is that these microorganisms, they will have the ability to produce a very strong immune response. Okay, how they are producing a strong immune response? Because since they are just weakened but not completely killed, they have the ability to proliferate a little bit inside the human body. Okay, when they proliferate inside the human body, our defense cells will identify it. Our immune cells will come and readily identify it and eliminate it. Okay, for a healthier individual. Okay, for a healthier individual, our healthy immune cells will come and identify this live organism that is present in our body inside the with the entered through the vaccine, and will try to eliminate. Okay, and when they encounter the same organism in future, then they will produce a very strong secondary response. Okay, so that's how live vaccines work. Then there is like inactivated vaccines, or in inactivated vaccines, we are completely killing the microbe. Okay, we are completely killing the microbe, and we are using it as vaccines. Okay, since the organism is like completely killed, they don't have the ability to proliferate inside the human body. Okay, it's like no proliferation capacity will be there. It's like completely killed. Okay, so the response will be good, but not as strong as the live vaccine. Okay, so it does require booster doses. Okay, in a particular time periods, we have to give the booster doses also. Okay, so that's about the inactivated vaccines. Then there is like subunit vaccine or the toxoid vaccine. Subunit vaccine means uh, the whole of the microbe is not used, only a particular protein or carbohydrate that is present on that surface of the antigen, that alone is used. Okay, so that's called as the subunit or the toxoid. Okay, since only a part of the microbe is used, here also no proliferation is happening inside the body. Okay, so it is also not as strong as live vaccine. So again, booster doses are required. Okay, so this is about the three types of vaccines. We have the live vaccine, inactivated vaccine, and the subunits or the toxoids. Okay, live vaccines are the ones which, when injected into the human body, they do like a very minimal proliferation inside the body. Okay, so these types of vaccines and how they act is extremely important, because questions can be asked like, an immunocompromised patient came for the vaccine, and which of the following vaccine is contraindicated for that immunocompromised patient? Okay, very commonly asked question. Okay, the one that was asked in the NEET MDS 2024 question paper also. Okay, uh, take it like an HIV patient or any other kind of immunocompromised patient comes to you for vaccination, and which of the following vaccine is contraindicated in such people? Okay, live vaccines will not be given for immunocompromised patients. Okay, live vaccines are always contraindicated for immunocompromised patients. Okay, why they are contraindicated? Because they have the ability for minimal proliferation inside the body. Okay, in a healthy individual, our healthy immune cells will come and attack them. Okay, but in an immunocompromised patient, they are already immunocompromised. Their immune cells are weak or not present. Okay, so these live vaccines, if you inject inside them, it will proliferate inside the body. Okay, there is no like cutback mechanism. Okay, there is no like negative mechanism to reduce that proliferation. Okay, there is no defense mechanism to reduce that proliferation. Okay, so that it will keep on proliferating and proliferate. Okay, so live vaccines are always contraindicated for immunocompromised patients. Okay, so which vaccines are included under these live vaccines? Tuberculosis vaccine, oral polio vaccines, measles, rotavirus, and yellow fever vaccines. Okay, these are all contraindicated, okay, for immunocompromised patients. Okay, remember that. Now, next we are moving on to antigens. Okay, so what are antigens? Antigens are any substance which, when introduced into the body, will help in the production of antibody. Okay, and it reacts in a very specific manner. Okay, so these antigens are any substance, okay, that when it comes in the inside the body, it will produce and it will help in the production of antibody. Okay, so such materials are called as antigens, and these antigens can be divided into three types. We have complete antigen. Complete antigens are capable of inducing antibody formation. Then there is something called as incomplete antigen or haptens. Okay, so these haptens are they does not induce this antibody formation. Okay, they are incapable of producing this antibody formation. Okay, then there is something called as super antigens. Okay, so the super antigens, they are the ones which overstimulate the immune system. Okay, uh, they are like some of the toxins from the staphylococcus and from the streptococci. What they does is that usually there is a antigen binding site that is present in the MHC molecule and in the T cell receptor. Okay, so that is called as the antigen binding site. Okay, so usually these antigens, they will go and bind at this site between the MHC and the T cell receptor. Okay, but what this super antigens does is that instead of binding at this particular site, they will go and bind at the lateral position like this. Okay, so when they go and bind at a lateral position like this, what happens is that these antigens will be activating lots of T cells. Okay, so they will be activating lots of T cells. These T cells will be like releasing so many cytokines, mainly the interleukin 2 also. So when this interleukin 2 is again and again released, what happens is that it will again leads to the like proliferation of many T cells. Okay, so overall the immune system will get overstimulated. Okay, so such antigens which are capable of overstimulating the immune system, they are called as super antigens. Okay, and the examples of super antigens include the enterotoxins that are released by staph and the pyogenic exotoxins that are released by the group A streptococci and mycobacterium tuberculosis, HIV, rabies virus, Epstein-Barr virus. These all have the capability to produce super antigens. Okay, so this is about the antigens and the different types of antigens. All right. Now, antigens, like I had already mentioned, they will help in the induction or the production of antibodies. Okay, so what are antibodies? They are like glycoprotein molecules. Okay, they are the ones that recognize this antigen and will facilitate the clearance of this antigen. Okay, so they are mainly present on the B cell membrane and they are secreted by the plasma cells. Okay, so if you look at the structure of this antibody, it will have two light chains. These red colored ones are the light chains and the lavender colored ones are the heavy chains. Okay, so an antibody, it will contain two light chains and two heavy chains. Okay, and at one end, there is an antigen binding portion. Okay, that is called as the FAB. Okay, so this is the antigen binding portion and this is the crystallizable portion. Okay, that is called as the FC. Okay, so these heavy chains and light chains, they are bonded together by the disulfide bonds. And at one end, we have the FAB portion and at the other end, we have the FC portion. Okay, so these heavy chains, they can be of different types. Okay, there is like very different types of heavy chains like gamma, mu, alpha, delta, and epsilon. Like that, so many different types of heavy chains are there. Okay, so based upon which heavy chain is present, there are 5 types of immunoglobulins. Okay, we have IgG, IgM, A, D, and E. Okay, so these are the 5 types of immunoglobulins. Now, out of these, E, G, M, and A are extremely important. Okay. So IgG, it is the major immunoglobulin that is present in the serum and it is the one with the longest half-life. Okay, 23 days. Okay, it has the longest half-life and it has the ability to cross placenta and even produce passive immunity for newborn. Okay, so the IgG is the one which crosses the placenta. Okay, when an antigen attacks our body, it is not the first antibody to come into place. Okay, IgG comes late only, but it will be present for a longer time. Okay, so it protects the microbes that is active in the blood and in the tissues. And there are four types also. That is like G1, G2, G3, and G4 are also there. Okay, so remember IgG, it is the major one and it has the longest half-life and it crosses placenta. Then we have the IgM. Okay, IgM is the heaviest immunoglobulin. Okay, it is also called as millionaire molecule. Okay, it is the largest and the most heaviest one. It is usually present in the intravascular region and it is the earliest immunoglobulin. Okay, it is the earliest immunoglobulin that is produced by the fetus also. Okay, within 20 days, we'll be able to see this IgM. Okay, and it does not cross placenta and it is very short-lived also. Okay, not as long-lived as IgG like 23 days and all. It is very short-lived, but its presence indicates that recently there was an infection present in the body. Okay, so that's about IgM, which is also called as millionaire molecule. Then next we have the IgA. Okay, so this IgA is the second major immunoglobulin. Its half-life is also less, like 6 to 8 days only. And there are two forms of IgA. There is serum IgA and the secretory IgA. Okay, so this serum IgA will be usually monomer structure, whereas secretory IgA will usually have a dimer structure like this. Okay, so this is a dimer structure. Two antibodies. It will be joined with the help of a J chain. Okay, so this J chain is also called as joining chain. Okay, so this J chain will be present only in two immunoglobulins. One is IgA and the other one is IgM. Okay, so only in these two immunoglobulins, you will be able to see this J chain. Okay. Mainly, the almost all the secretions contain this IgA and it protects the mucous membrane also. Then IgE. Okay, IgE, it resembles IgG and its half-life is just 2 days and it is the only heat labile immunoglobulin. Okay, the only heat labile immunoglobulin. Okay, it does not cross a placental barrier or it usually has an affinity for mast cells. Okay, it is usually present in the mast cells and it is the one which like produces this anaphylactic reaction. Okay, against allergens. All the anaphylactic reactions are mainly produced by this IgE only. Okay, and it is the only heat labile immunoglobulin. Okay, so this is about all the major immunoglobulins. Now, this uh, table like summarizes all the immunoglobulins and their characteristic feature. Okay. Now, next is the hypersensitivity reaction. Let's have a look at the hypersensitivity reaction. There are like four types of hypersensitivity reaction, and we can broadly classify them into immediate hypersensitivity reaction and delayed hypersensitivity reaction. Okay, so this immediate hypersensitivity reactions, they are usually mediated by B cells or antibodies, and it includes the type 1, type 2, and type 3. But the delayed type, which is mediated by the T cell, it is called as the type 4 or the T cell mediated hypersensitivity reaction. Okay, so in type 1 hypersensitivity reaction, which is also called as allergy. Okay, so what is mainly happening is that if the allergen is like inhaled or ingested, or if it is the person is like getting infected with that allergen, it will be first recognized by the dendritic cells. Okay, so it will be first recognized by the dendritic cells. these dendritic cells are like antigen presenting cells. Okay, so they will take that antigen and go and present it to the T cells. Okay, it will because of the presence of this presentation of this antigen by the dendritic cell, this T cell, it will get activated and it will produce the Th2 cells, okay, T helper cells, and it will secrete like lots of cytokines. Okay, which all cytokines can be released? There will be like interleukin 4, interleukin 5, interleukin 13, all these interleukins can be released. Okay, so this interleukin 4, it mainly stimulates the B cells. Okay, and interleukin 5, it activates the eosinophils, and interleukin 13, it activates on the epithelial cells. Okay, and it stimulates the mucus secretion. Now, like if there is like repeat exposure of the same allergen, then what happens is that these mast cells, they are like already exposed to this antigen. Okay, so what happens is that the it will release its own mediators. Okay, it will release mediators like the vasoactive amines and lipid mediators will be released, and the cytokines will also be released. Okay, so these histamines are the one which causes like vasodilation, increased vascular permeability, smooth muscle contraction, and all those will be seen. Okay, intense bronchospasm by these prostaglandins and the increased mucus secretion will all be done with the help of these mast cell degranulation. Okay, so this hypersensitivity reaction can be of two types. We have an acute reaction and a chronic reaction. The acute reaction is called as the anaphylaxis, and the chronic reaction, it is called as atopy. Okay, any food allergy, bee sting allergy, or the drug allergy, they are all examples of this type 1 hypersensitivity reaction. Okay, the bronchial asthma, the allergic rhinitis, or the sinusitis, and all kinds of food allergies are all examples of this type 1 hypersensitivity reaction. Okay, now let's have a look at the type 2 hypersensitivity reaction. In the type 2 hypersensitivity reaction, it is mainly mediated by immunoglobulin G and or rarely immunoglobulin M. Okay, so what it does is that it will go and bind to the antigen that is present on the target cell or the tissue. Okay, so these antigens, it will be first opsonized. Okay, opsonized is like making the antigen more tasty for the defense cells. Okay, so that is done by the process called as opsonization. Okay, so this opsonization the cell will be opsonized, and this antigen will be binded to the antigen on the target cell or the tissue. Okay, when it binds, there will be phagocytosis happen. Okay, it will try to ingest the organism and try to phagocyte. Okay, then at the particular site, more and more leukocytes will also be recruited so that all the systemic signs and symptoms will be shown. Okay, so all the diseases like the hemolytic anemia or the thrombocytopenic purpura, pemphigus vulgaris, rheumatic fever, myasthenia gravis, and the pernicious anemia are all examples of this type 2 hypersensitivity reaction. Okay, coming to type 3 hypersensitivity reaction, which is like immune complex mediated. Okay, immune complex mediated means the antigen antibody complexes are formed. Okay, and these complexes are like activating the complement system, and there will be like recruitment of these leukocytes by the complement products and the FC receptors also. So when these leukocytes and these complement products are like activated, it will release lots of enzymes and other toxic molecules. Okay, so that will lead to type 3 hypersensitivity reaction, which is mainly shown in SLE. Okay, systemic lupus erythematosus, in polyarteritis nodosa, in reactive arthritis, serum sickness, and Arthur's reaction. These are all examples of this type 3 hypersensitivity reaction. Okay. Then the last one, that is the type 4 hypersensitivity reaction, which is usually T cell mediated or delayed hypersensitivity reaction. Okay. So in this delayed hypersensitivity reaction, what is happening is that these T lymphocytes, they will release lots of cytokines, which will result in inflammation and macrophage activation. Or these T cells, they can directly go and attack. Okay. The CD8 cells, they will directly go and attack and will lead to cell killing and the tissue injury. Okay. So that are the two types of type 4 mediated hypersensitivity reaction. Okay. With the help of cytokines, they can cause injury to the tissue, or the cells itself can directly go and kill the CD8 cells. They itself can directly go and kill the normal cells. Okay, so this is mainly seen in the type 4 hypersensitivity reaction. Examples are mainly rheumatoid arthritis, multiple sclerosis, type 1 diabetes mellitus, psoriasis, contact sensitivity, IBD, okay, inflammatory bowel disease. All these are examples of this type 4 hypersensitivity reaction. Okay, so this is all the types of hypersensitivity reaction in a gist in a table. Please go through this table for your revision time also. Okay, it will be much easier for you, okay, to read the table instead of reading the entire text. Okay, so this is regarding the some of the important concepts that are present in the initial chapters of the microbiology, including general microbiology and the immunology. Now let's discuss some of the questions from the same topics. Okay, so, like I already used to say, if you know the answer after reading the question and the options, if you know the answer, you can put it in the chat box. Even if the answers are wrong, it doesn't matter. Okay, we are all in a learning phase now. Okay, so make mistakes now. Okay, so that you don't make it in the exam hall. Okay, so let's have look at the first question. All are sporicidal agents except, is it glutaraldehyde or formaldehyde or ethylene oxide or ethyl alcohol, which is not a sporicidal agent. Sterilization is the method of yes, killing even the spores. But yes, there are chemicals which can be used just for antiseptic and disinfectant only. They are not sporicidal agents. Yes, Dr. Kartika, you are right. It is ethyl alcohol. Okay, ethyl alcohol is not a sporicidal. Okay, it is just an skin antiseptic. Okay, not a sporicidal agent. Now, next question. The ideal method of sterilization of pre-packed plastic syringe. Okay, which is the ideal method of sterilization of pre-packed plastic syringe. Okay, since it's plastic, it cannot be exposed to high temperatures of heat. Okay, so which one of the following is the correct method of sterilization? Yes, it is infrared radiation. Okay, we usually use the infrared radiation for this pre-packed plastic syringe. Okay, it is like a cold sterilization method where we are not the temperature of the sterilized item is not increased. So it is a method of cold sterilization, which we use for plastic syringes. Okay, the heat sensitive items like the pre-packed plastic syringes, we mainly use infrared radiation. Okay. Now, next question. Find out the correct statement regarding the sterilization control. Okay. So, the spores of Bacillus stereothermophilus is used as biological indicator of plasma sterilization, or whether it is used as biological indicator of autoclave, or whether it is used as biological indicator. Bacillus atrophius is used as biological indicator of ionizing radiation, or Bacillus atrophius is used as biological indicator of ethylene oxide sterilization. Which is the correct answer? Yes. Only 1, 2, and 4 are right. Okay. Bacillus stereothermophilus is used for plasma sterilization. That is true. Bacillus stereothermophilus is used as for autoclave. That is also true. Bacillus atrophius is used for ethylene oxide sterilization. That's also true. But for ionizing radiation, we usually do not use Bacillus atrophius. Okay. Bacillus atrophius is not used as a biological indicator of ionizing radiation. Okay. Then the next question is McConkey's medium is. What is McConkey's medium? Is it a differential media or enriched media or enrichment media or selective media? Okay, so like I had already mentioned, this is a NEET MDS 2024 question. Yes, it is a differential media. Yes, you are right, Dr. Kartika. So next question, choose the anaerobic culture media among the following. Is it alkaline peptone water or thioglycolate medium or Robertson's cooked meat medium or selenite F broth? Which among the four is a anaerobic culture media? Like we had already discussed, thioglycolate medium is an anaerobic culture media and Robertson's cooked meat medium also. Okay, so the correct answer is option B, 2 and 3. Okay. Remember the uh, aerobes growing at the top and the anaerobes going at the bottom for the thioglycolate media. Now, the tissues given for IHC are transported in. Okay, this is another NEET MDS 2024 question. So, which is the transport media for the tissues that are indicated for immunohistochemistry? Is it normal saline or water or alcohol or Michel solution? Yes, Dr. Kartika, you are right again. It is Michel solution. Okay, because it preserves the immunoantigenicity of the tissues. All right. Now, the immunoglobulin that mediates primary immune response is. Which of the immunoglobulin is the one that mediates the primary immune response? Is it A, E, or M, or G? The primary one is option C. It's IgM. Okay, IgM is usually the first one to appear. Okay. G, I told you, it's the last one to appear, but it is long living. Next, the antibody that is responsible for anaphylactic reaction. Okay, very easy. Which is the antibody that is responsible for anaphylactic reaction? Yes, you're right again, Dr. Kartika. It is IgE. Okay. Now, the following statements are true about super antigens, except they are bacterial proteins. They are antigens that are present on the surface of Bombay group RBCs. They cause a polyclonal T cell activation. They cause syndrome similar to septic shock. Okay, which of the following statement are false? Okay. You should be really careful when they use words like this, except. Okay, the following statements are true about superantigens, except. Okay, so they are referring to a wrong statement among the following or a false statement among the following. Okay, so which among the following is false? Are they bacterial proteins? Yes, they are bacterial proteins. Does they cause T cell activation? Yes, they cause like immense polyclonal T cell activation by this release of IL-2 and all. They cause syndrome similar to septic shock? Yes. Okay. But are they the antigens that are present on the Bombay group RBCs? No. Okay, this is wrong. Okay, they are not the antigens on surface of Bombay group RBCs, but they are bacterial proteins. Okay, they can be seen in streptococcal toxins or staphylococcal toxins or in mycobacteria or HIV like that. They are not the antigens that is present on the RBCs. Okay, so B is the answer. Now identify the structure shown in the image below. Okay, which structure is that? Pentamer structure. This is called as the pentamer structure. Okay, pentamer structure means like this, 5 fingers. Okay, pentamer structure along with a G chain. Okay, only 2 immunoglobulins have G chain, and only one immunoglobulin shows this pentamer structure. Yes, it is IgM, which is also called as the millionaire molecule. Okay, it is the most heaviest immunoglobulin. Now, which of the following vaccine is contraindicated in immunocompromised patients? Okay, another NEET MDS 2024 question. Okay, which is contraindicated in immunocompromised patients? Like I had already mentioned, live vaccines are contraindicated in immunocompromised patients. Okay, which among this is a live vaccine? DPT? No. Killed influenza vaccine, it itself has killed in it. Okay, no. Pneumococcal vaccine also? No. The only live vaccine that is present among the following is oral polio vaccine. So polio vaccines are usually contraindicated in immunocompromised patients. Now next, a 6-year-old child develops a rapid onset of urticaria, lip swelling, wheezing, and difficulty breathing within minutes after consuming peanuts. Which type of hypersensitivity reaction does this represent? Okay. Typical case of food allergy. Okay, food allergy to peanuts. Okay, all the symptoms like urticaria, lip swelling, wheezing, difficulty breathing, and all are present within minutes after consuming peanuts. Okay, so which type of hypersensitivity reaction does this represent? Which type, under which type the food allergy comes? It's under type 1. Okay, so it is a type 1 hypersensitivity reaction. Okay, so they can also ask such clinical based questions also. Okay, you must be prepared for that. Okay, just look at the key points that are present in the question. Okay, so by reading the first line, first sentence itself, we can see that this is a typical case of food allergy. Okay, so which hypersensitivity reaction indicates towards food allergy? Type 1 hypersensitivity reaction. Okay. So that's all for today. We will meet for the second class of microbiology 2 days later. Mainly, we'll be discussing in detail about bacteriology. Okay, important bacteria and the medium that is used to grow this bacteria and some of the important reactions that are exhibited by bacteria. We'll be discussing all those in detail. Okay, so please have a look at those portions. Okay, just go through that portion alone. Okay, and if you have any doubt, you can ask me now. Any doubts related to immunology and this general microbiology chapters? Okay, Dr. Ratika. Thank you so much for attending the session. We'll see you for the next session. And if you have any doubt, you can DM me personally or you can ask in the WhatsApp group also. No issues. We'll definitely answer all your queries. And if you have, and you can also comment below this video also. Okay, no issues. We will definitely answer all your queries. Okay. So yes, thank you so much. Good night and goodbye.